All right, so today I'm going to introduce our very first lab that we will be using argument-driven inquiry for. So this one is titled Osmosis and Diffusion. Why do red blood cells appear bigger after being exposed to distilled water? So in this video, I want to read through the introduction and start giving you some ideas of how to set up your lab. So go ahead and follow along with me. All living things are made of cells. Some organisms, such as bacteria, are unicellular, which means they consist of a single cell. Other organisms, such as humans, fish, and plants, are multicellular, which means they consist of many cells. All cells have some parts in common. One part found in all cells is the cell membrane. The cell membrane surrounds the cell, holds the other parts of the cell in place, and protects the cell. Molecules such as oxygen, water, and carbon dioxide can pass in and out of the cell membrane. All cells also contain cytoplasm. The cytoplasm is a jelly-like substance inside the cell where most of the cell's activities take place. It's made out of water and other chemicals. Some cells found in multicellular organisms are highly specialized and carry out very specific functions. An example of a specialized cell found in vertebrates is the erythrocyte, or red blood cell. Red blood cells are by far the most abundant cells in the body. The primary function of red blood cells is to transport oxygen from the lungs to the cells of the body. In the capillaries, the oxygen is released so other cells can use it. 97% of the oxygen that is carried by the blood from the lungs is carried by hemoglobin. The other 3% is dissolved in the plasma. Hemoglobin allows the blood to transport 30 to 100 times more oxygen than could be dissolved in the plasma alone. As you can see in the figure to the right, red blood cells look like little discs when they are viewed under a microscope. They have no nucleus. The nucleus is extruded from the cell as it matures to make room for more hemoglobin. A unique feature of red blood cells is that they can change shape. This ability allows them to squeeze through capillaries without breaking. Red blood cells will also change shape in response to changes in the environment. For example, if you add a few drops of distilled water to blood on a microscope slide, the cells will look bigger after a few seconds. See the figure's right panel. So this picture right here. So before you add water, here's what they look like, and after they look much larger. Scientists often develop and test explanations for natural phenomena. In this investigation, you will have an opportunity to design and carry out an experiment to test two different explanations of why red blood cells appear bigger after they are exposed to distilled water. These are the two explanations you will test. The first explanation is molecules such as protein and polysaccharides are more concentrated inside the cell than outside the cell when the cell is in distilled water. These molecules therefore begin to move out of the cell because of the process of diffusion, but are blocked by the cell membrane. As a result, these molecules push on the cell membrane and make the cell appear bigger. The second explanation is water molecules move into the cell because the concentration of water is greater outside the cell than it is inside the cell. As a result, water fills the cell and makes it appear bigger. Your task. Your task is to design and carry out an experiment to determine which of the two explanations about the appearance of red blood cells after exposure to distilled water is the most valid or acceptable from a scientific perspective. The guiding question of this investigation is, why do red blood cells appear bigger after being exposed to distilled water? Okay, so what this means is, you have to answer this question. Why do the red blood cells appear bigger after being exposed to distilled water? And I'll give you a hint. One of these explanations right here is the correct explanation. So you are going to design your own experiment to test both of these explanations, and then you'll be able to come to the conclusion which one is actually the correct answer. So here are the materials that'll be available to you this upcoming week. There's starch solution, which is a polysaccharide, distilled water, beakers, graduated cylinders, a balance, electronic or triple beam, depending on which classroom you're in, dialysis tubing, safety goggles, and aprons. Before you begin the lab, there are some safety precautions to consider. So one, indirectly vented chemicals splash goggles and aprons must be worn. Wash your hands with soap and water after completing this lab and follow all normal lab and safety rules. All right, so getting started. You will use models of cells rather than real cells during your experiment. You will use cell models for two reasons. One, a model of a cell is much larger than a real cell, which makes the process of data collection much easier. And two, you can create your cell models in any way you see fit, which makes it easier to control for a wide range of variables during your experiment. The cell models will therefore allow you to design a more informative test of the two alternative explanations outlined above. You can construct a model cell by using the dialysis tubing. Dialysis tubing behaves much like a cell membrane. To create a model of a cell, place the dialysis tubing in water until it is thoroughly soaked. Remove the soaked tubing from the water and tightly twist one end several times and either tie with string or tie a knot in the tubing. You can then fill the model cell with either a starch solution or distilled water. Once filled, twist the open end several times and tie it tightly as shown in the figure. You can then dry the bag and place it into any type of solution you need. So here, I know it's hard to see, but here is your dialysis tubing. You can close off the bottom end, and then once you fill it, you close off the top end. You can either tie a knot in these, or I also have clips. In the next video, I will do some tool talk, and I'll show you how to put the clips on. In designing your experiment, you must determine what type of data you will need to collect, how you will collect it, and how you will analyze it. To determine what type of data you will need to collect, think about the following questions. What will serve as your dependent variable? Meaning, is it going to be the mass of the cell or the size of the cell? What are you going to measure? Is it going to be easier to get the mass and just put your sample on a balance? Or are you going to somehow measure like the circumference using maybe string and a ruler? What type of measurements will you need to make during your investigation? Again, are you going to go for like mass? Are you going for grams? Are you going to go for diameter and maybe measure either millimeters or centimeters? To determine how you will collect your data, think about the following questions. What will serve as a control or comparison condition? How do you know if you're putting a starch in your bag or your solution? How do you know that it's making any difference than just distilled water alone? What types of treatment conditions will you need to set up and how will you do it? How many cells will you need to use in each condition? How often will you collect data and when will you do it? How will you make sure your data are of high quality? Meaning, how will you reduce error? How will you keep track of the data you collect and how will you organize the data? To determine how you will analyze your data, think about the following questions. How will you determine if there is a difference between the treatment conditions and the control condition? What type of calculations will you need to make? What type of graph could you create to help make sense of your data? All right, for this lab, investigation proposal required? No, but you are more than welcome to run your ideas by me before you begin. All right, next, part of argument-driven inquiry and its strengths is it does do a really good job of connecting cross-cutting concepts and bringing in the nature of science into our labs. So this section is on connections to cross-cutting concepts into the nature of science and the nature of scientific inquiry. As you work through your investigation, be sure to think about the importance of identifying the underlying cause for observations, how models are used to study natural phenomena, how matter moves within or through a system, the difference between data and evidence in science, and the nature and role of experiments in science. All right, after you collect all of your data, we will enter the argumentation session. Once your group has finished collecting and analyzing your data, prepare a whiteboard that you can use to share your initial argument. Your whiteboard should include all the information shown in the figure below. And for us, instead of a whiteboard, we will use either a PowerPoint slide or a Google slide just on your computer screen. So on your slide, you will have the guiding question, your claim, the evidence you use in your claim, and how you justify that evidence. To share your argument with others, we will be using a round-robin format. This means that one member of your group will stay at your lab station to share your group's argument, while the other members of your group go to the other lab stations one at a time to listen and to critique the arguments developed by your classmates. The goal of the argumentation session is not to convince others that your argument is the best one. Rather, the goal is to identify errors or instances of faulty reasoning in the arguments so these mistakes can be fixed. You will therefore need to evaluate the content of the claim, the quality of the evidence used to support the claim, and the strength of the justification of the evidence included in each argument that you see. In order to critique an argument, you will need more information than what is included on the whiteboard. You might therefore need to ask the presenter one or more follow-up questions, such as, how did you collect your data? What did you use? Why did you use that method? Why did you collect those data? What did you do to make sure the data you collected are reliable? What did you do to decrease measurement error? What did you do to analyze your data? Why did you decide to do it that way? Did you check your calculations? Is that the only way to interpret the results of your analysis? How do you know that your interpretation of your analysis is appropriate? Why did your group decide to present your evidence in that matter? What other claims did your group discuss before you decided on that one? Why did your group abandon those alternative ideas? How confident are you that your claim is valid? What could you do to increase your confidence? Once the argumentation session is complete, you will have a chance to meet with your group and revise your original argument. Your group might need to gather more data or design a way to test one or more alternative claims as a part of this process. Remember, your goal at this stage of the investigation is to develop the most valid or acceptable answer to the research question. Report. Once you have completed your research, you will need to prepare an investigation report that consists of 3 sections that provide answers to the following questions. 1. What question were you trying to answer and why? 2. What did you do during your investigation and why did you conduct your investigation in this way? 3. What is your argument? Your report should answer these questions in 2 pages or less. This report must be typed, and any diagrams, figures, or tables should be embedded into the document. Be sure to write in a persuasive style. You are trying to convince others that your claim is acceptable or valid. Okay, so I know that seems like a lot, but we will work on this all week in class. Okay, so we will have a lot of class time to do this. So today, while you're watching these videos at home, is just to introduce argument-driven inquiry. And to start getting some ideas about what kind of method you're going to follow to answer your guiding question, which is, why do the red blood cells appear bigger after being exposed to distilled water? And then the next video you're going to watch is Tool Talk, and I will demonstrate how to actually make your cell models and some techniques, which will also help you brainstorm some ideas and help you make that procedure.